Hi guys, my name is Jamil Ahmad and we were discussing about the transcription. Welcome back. Today we will discuss about another topic in the biochemistry, and this is about the ribosomal RNA used to construct ribosomes. So mean that what is the role of ribosomal RNA in the construction of the ribosomes. So to learn about this knowledge, to learn about this topic, first we must know about ribosomes, that what are the ribosomes. I would like to give you some concepts about ribosomes. Ribosomes are the membraneless organelles. These are the membraneless organelles. These are the membraneless organelles inside the cell. These are the cell organelles that are membraneless, and these are produced, or we can say that formation of ribosomes, synthesis of ribosomes happens inside the nucleolus of nucleus, nucleus. Okay, and then after the formation, they move out from nucleus to cytoplasm, and they have their location of two types. If we talk about the location, then they have two types of location. Either they are floating in the cytoplasm, or we can say that second one, that they are fixed on the rough endoplasmic reticulum. And if we talk about the function of ribosomes, the function of ribosomes is this, that the ribosomes are the factory of proteins. As you know that formation of proteins happens with the help of ribosomes. So I would like to repeat this concept, very simple, that ribosomes are the membranous organelles which are produced or synthesis inside the nucleus of nucleus, and after the formation this ribosomes moves outside from nucleus to cytoplasm, and it has two kinds of location. One is maybe floating in the cytoplasm, second they are fixed on the rough endoplasmic reticulum. And then what is the function of ribosomes? The function of ribosome is the factory of proteins. So I would like to tell you two MCQs. It's bridge to physiology MCQs, but I would like to tell you in this topic as well that ribosomes is the factory of proteins, and and nucleolus, or we can say the nucleus, is the factory of ribosomes. Mean that ribosomes are synthesized inside the nucleus, but proteins are synthesized by ribosomes. So ribosomes are the factory of proteins, and nucleolus is the factory of ribosomes. So this was about the ribosomes. The second thing, that ribosomes are made up of two things. The first one is the ribosomes is made up of two things. The first one is ribosomal RNA plus ribosomal proteins. So here is a question, that ribosomes are made up of two things. The first one is the ribosomal RNA, and second is the ribosomal proteins. So here arises another question, that what is the ribosomal RNA? As you know that RNA, ribonucleic acid, has three types of RNA. Many types of RNA, but most important, most famous are three types: transfer RNA, ribosomal RNA, messenger RNA, heterogeneous nuclear RNA, that is a pre-messenger RNA, and there is also small nuclear RNA. So many types of RNA. So ribosomal RNA, this is one of the types of the RNA, ribonucleic acids, and this RNA is the most abundant type of RNA. Ribosomal RNA is most abundant type of RNA. This is the most abundant type of RNA. It is almost 80% of total RNA, total RNA. So this is the most abundant type of RNA, or we can say that it is the 80% of total RNA. So we can say that this is the most abundant type of RNA. And second thing, that about this ribosomal RNA is the most, we can say the stable form of RNA. This is the most stable form of RNA. This is the second concept. Second feature about the ribosomal RNA. And if you talk about third one, then the third one, these ribosomal RNA associates with these ribosomal proteins, and they form some kind of machinery structure, and they will form the machinery structure. Okay, machinery structure, and this machinery structures on the surface of this machinery structures, transfer RNA and messenger RNA comes, and they interacts with each other, and they cause the translation. And important for the synthesis of proteins. So this is a simple concept that ribosomal RNA is the most abundant type of RNA. It contains 80% of the RNA, number one. And number second concept, that most stable form of RNA. And third one is that ribosomal RNA associates with these ribosomal proteins. So ribosomal RNA and ribosomal proteins will associate with each other, and they will form the machinery structure. That machinery structure is the ribosomes. So they will form the machinery structure that is called the ribosomes. Then on the surface of this machinery structure, on the surface of this, on the structure of these ribosomes, 2 and the types of RNA will come, that is the transfer RNA and messenger RNA. They will interact with each other, and they will cause the translation, and in the result of translation, proteins will form. Synthesis of proteins will happen. As I have said earlier, the ribosomes is the factory of proteins. So in this way, proteins will formed. Okay, so now we will move on, and we will see that ribosomes are prokaryotic ribosomes as well and eukaryotic ribosomes. Two type of ribosomes, prokaryotic ribosomes and eukaryotic ribosomes. And another question is that how ribosomal proteins and ribosomal RNA will combine with each other, and how they will form the ribosomes, and they are different in prokaryotes and different in eukaryotes. First we are going to see the prokaryotic ribosomes, then we will talk about the eukaryotic ribosomes. So first one is the prokaryotic ribosome. So most important, you have to learn about it, that prokaryotic ribosome is of 70S. Okay, prokaryotic ribosome is the 70S, and this 70S further consists of the two subunits. The one is the larger subunit, so this one is the larger subunit, 50S, larger subunit, and second is the smaller subunit, that is a 30S. So what is the concept? The concept is this, that the prokaryotic ribosomes is of 70S, and this 70S is made up of two further subunits, that is the larger subunit and smaller subunit. Larger subunit is of 50S and smaller subunit is of 30S. So 50S plus 30S will form the 70S, that is the prokaryotic ribosomal RNA. Okay, so larger subunit and smaller subunit. Larger subunit is of 50S and smaller subunit is of 30S. These larger and smaller subunits will combine with each other, and they will form this 70S, that is the prokaryotic ribosome. Okay. So second thing, I would like to give you a little bit concept that how can you remember that prokaryotic ribosomes is 70S, and here you can see that eukaryotic ribosomes are of 80S, 60S, 40S. How you can learn these numbers, how you can remember these numbers, because it may be difficult, it may be mixed up with the eukaryotic ribosomes. So remember one concept, that these prokaryotic ribosomes will always be the odd numbers. These will be the odd numbers. These will be the odd numbers. You can see that in 70, the first one, 7 is the odd, and 5 is the odd, and 3 is the odd. So this is the concept to remember the prokaryotic ribosomes. So 70 as 7, 5, 3. So these are all are the odd numbers. So two concepts. The first one is that 70S made up of two further subunits. One is the larger subunit, that is the 50S, and smaller subunit, that is the 30S. They will combine with each other, and they will form the 70S subunit, and we can remember these by the odd numbers. And the next one, now we will talk about that further in detail, that this larger subunit, that is the 50S subunit, made up of two things. You know that ribosome. This is the ribosome, 50S ribosome subunit, that is made up of ribosomal RNA and ribosomal proteins. Okay, ribosomal RNA and ribosomal proteins. So look at this. Look at this one. This is the this 50S subunit consists of the further 2 units, that is the 5s ribosomal RNA and 23s ribosomal RNA plus 34 proteins. So larger subunit further made up of 2 ribosomal RNAs and 34 proteins. 2 ribosomal RNAs are 5s RNA and 23s RNA and 34 proteins. These will combine with each other and they will form the larger subunit of the ribosomes. As I have said earlier here that ribosomes are made up of ribosomal RNA plus ribosomal proteins. So here ribosomal RNA larger subunit, 2 ribosomal RNA and 34 proteins. Okay. Now smaller subunit is of 30s and it is further made up of 16s ribosomal RNA and 21 proteins. So it contains 21 proteins and 60s ribosomal RNA. Okay, but this is not too much important to remember. The important concept you have to remember that is it is 70s, 50s, 30s. Okay, this is important. Not too much important to learn that this is the 5s, 23s, 16s, 21 proteins, 34 proteins. It's not too much important, but you have to learn these for basic concepts. And we move in the detail. Then these both you know that the 50s subunit, this is the 50s subunit and this is the 30s subunits. These both are the subunits. These both subunits are made up of the same RNA polymerase. You know that prokaryotic polymerase is a one polymerase, that is the RNA polymerase alpha 2 beta prime. So these all, either it is a larger subunit or small subunit, either it is a 5s ribosomal RNA or 23s ribosomal RNA, or we can say that 16s ribosomal RNA, or these ribosomal RNA are produced by the RNA polymerase alpha 2 beta prime. So alpha 2 beta prime will act on the DNA and it will form the all kinds of these ribosomal RNA. Okay. So now we will move on. In the next step, we will see that the thing most important you have to remember here that what is the role of 16s ribosomal RNA. 16s ribosomal RNA goes and it recognizes the Shine-Dalgarno sequence, and it binds with the Shine-Dalgarno sequence and causes the initiation of translation. So first you must know that what is the Shine-Dalgarno sequence. Remember in the prokaryotic transcription, the messenger RNA contains Shine-Dalgarno sequence. Where are the Shine-Dalgarno sequence? Let's suppose that this is the messenger RNA. Then you know that let's suppose this is a 5 prime and this is the 3 prime and this is the messenger RNA. And this messenger RNA contains some untranslated regions. Let's suppose that this is the untranslated region, this is the untranslated regions. So on the messenger RNA in prokaryotes there are some regions that are untranslated regions. So this Shine-Dalgarno sequence is the 5 prime untranslated region on the messenger RNA. So it is towards here, it will be sequence, it will be something like that. So let's suppose that this is the Shine-Dalgarno sequence on the messenger RNA, that is on the 5 prime untranslated regions of the messenger RNA there is a Shine-Dalgarno sequence. So this messenger RNA will come and this Shine-Dalgarno sequence and this 16s ribosomal RNA will recognize this Shine-Dalgarno sequence and they will bind with each other. So in this way this messenger RNA will land on the surface, or we can say that will connect with the ribosomes in this way. So this messenger RNA will come and it will land on the surface of this machinery structure. In this way the Shine-Dalgarno sequence will connect with 16s ribosomal RNA and they will cause the initiation of translation. Okay. So not too much worried about this concept, just things you have to remember are such few things. First thing that prokaryotic ribosomes are of 70s, 50s larger, 30s smaller, and another thing you have to learn that this 16s ribosomal RNA is used to recognize the Shine-Dalgarno sequence for initiation of translation. And another concept you have to remember, that is this concept that these are ribosomal types are produced by the RNA polymerase alpha 2 beta prime. In that same RNA polymerase will form all the types of these ribosomes. Okay. Now I think that prokaryotic ribosomes is okay for you. Now we will move on and we will look at the eukaryotic ribosomes. So as you see that in this case prokaryotic ribosomes were the 80s, 70s, but here eukaryotic ribosomes is of 80s. Okay, here is 70s, here is 80s, and it is further consists of the two subunits, that is the larger subunit and smaller subunit. So further two units, first one is the larger subunit and smaller subunit. Okay, in this case the larger one is of 60s and smaller one is of 40s. So this larger and smaller 60-40 will combine with each other, this 61 and 41 will combine with each other, and they will form the 80s ribosomal they will form the 80s ribosomes. So larger subunit is 80s and smaller is of 40s, and they will form the 80s ribosomes, that is the eukaryotic ribosomes. And another concept to remember these as well, that you can remember the eukaryotic ribosomes by even numbers. In case of prokaryotes, that was the odd numbers 7, 5, 3, but here is the even numbers 8, 6, 4. So all are the even numbers. So okay, I think now this concept is clear for you. Now we will move on and we will see that because ATS consists of two units, larger and smaller, and larger unit is further consists of 5s ribosomal RNA, 5.8s ribosomal RNA, and 20s ribosomal RNA plus 50 proteins. Okay, so this largest subunit further consists of the 5s ribosomal RNA, 5.8s ribosomal RNA, 28s ribosomal RNA plus 50 proteins. And if we talk about the smaller subunit, is further consists of the 18s ribosomal RNA and 30 proteins. I will say once again that it's very tough to remember all of these, not too much, but it's tough. So please don't bother about that you have to remember in any ways, no, don't worry about it. This is just for the basic concept. The main thing you have to remember that just this is 80s, larger one is 60s, and smaller one is of 40s. Okay, this is okay. And next thing that now I would like to say you something about that. Now you can see here that this 5.8s 8s ribosomal RNA, 28s ribosomal RNA, and 18s ribosomal RNA, these three types of ribosomal RNA in eukaryotes is produced by the RNA polymerase 1. RNA polymerase 1. So RNA polymerase 1 will synthesize the 5.8s ribosomal RNA, 28s ribosomal RNA, and 18s ribosomal RNA. So these three ribosomal RNA are produced by the RNA polymerase 1, and this RNA polymerase 1 will synthesize these three ribosomal RNA not in pieces. First it will synthesize the 45s ribosomal RNA as a one piece. RNA polymerase 1 first will synthesize the 45s ribosomal RNA, then this 45 ribosomal RNA will be cleaved into different pieces, and these pieces are the 5.8s ribosomal RNA, 28s ribosomal RNA, and 18s ribosomal RNA. And here remains just one ribosomal RNA, as you can see that 5s ribosomal RNA. Then this 5s ribosomal RNA is produced by the RNA polymerase 3. Okay, so these concepts is very important. You have to remember these concepts, that 5.8s ribosomal RNA, 28s ribosomal RNA, and 18s ribosomal RNA is produced by the RNA polymerase 1 as a single piece, that is a 45s ribosomal RNA. Then this single piece will cleave into different pieces in these different pieces, and 5s ribosomal RNA of eukaryotic ribosome is produced by the synthesized by the RNA polymerase 3 enzyme by a separate gene, by a different gene. Okay, so now we will move on. Same like that 16s recognize the Shine-Dalgarno sequence for initiation of translation. In this way, this 18s ribosomal RNA recognize the 7-methyl guanyl cap, and cap of messenger RNA, and causes the initiation of translation. You know that as we discussed in the previous lectures, that let's suppose that if this one is the messenger RNA that is produced from the eukaryotic transcription, you know that in eukaryotic transcription the messenger RNA produced is not a mature messenger RNA, it is a immature messenger RNA. Then after that capping happens, 7-methyl guanine cap, added, then A is poly A tail is added, then after that splicing happens. During the splicing, introns will be removed outside and exons will join together. About this I have explained in the previous lectures as well, but here is just a little bit I repeat. Then after that it will be changed into mature messenger RNA. So this is the messenger RNA, and this messenger RNA has 7-methyl cap as well. Then this 18S ribosomal RNA of ribosomes will attach with this 7-methyl G cap, and after the attachment there will be the initiation of translation, and after the translation proteins will form. As I have said here as well that these ribosomal RNA and ribosomal proteins will form the ribosome, and on this ribosome the messenger RNA will come, transfer RNA will come, and they will attach on this machinery structure, and they will cause the translation and they will form the proteins. So in this way they will form the proteins. Once again I would like to say you that don't bother to learn everything, but just learn few concepts that is very important. But you can learn all for good study, but you have to learn some basic concepts very well. That is the eukaryotic ribosomes that have 80S and it is composed of 2, larger is the 60S, smaller is the 40S. This is the one concept. And second concept is that you have to remember that 5.8S ribosomal RNA, 28S ribosomal RNA and 18S ribosomal RNA are produced by the RNA polymerase 1 as a single piece of ribosomal RNA, that is a 45S ribosomal RNA. Then this 45S ribosomal RNA will be cleaved into different pieces. Okay. And second concept that 5S ribosomal RNA is produced by the RNA polymerase 3 by a separate gene. Okay. And another concept you have to remember that 18S ribosomal RNA recognizes the 7-methyl G cap for the initiation of translation. So these were some concepts about eukaryotic ribosomes and prokaryotic ribosomes, difference between both. Next one is here I am using the one thing, it must be very different for you, that there is a S, I am using S and everything S, and you should focus on one point, that you should feel one point, that 50 plus 30 it must be 80, but here is a 70, and same like that 60 plus 40 must be 100, but here is a 80, and same like that. You can see that if we are going to add these, then they are not going to become the 45, but here is these are 45. So what is the reason behind that? So this S is not equal to molecular weight. This is not a molecular weight. So this S is not a molecular weight. This S is not a molecular weight. Rather, this S is the sedimentation coefficient. Sedimentation coefficient, it is a Swedberg unit. It is a Swedberg unit. What does that mean? It does mean that it is the behavior of particles. This S Swedberg unit, or we can say that sedimentation coefficients shows the behavior of particles, particles in centrifuge machine. So this is the behavior of particles in centrifuge machine. When centrifuge machine will spin, then what is the behavior of particles? This behavior is S. This behavior is S. It is not a molecular weight. This is the behavior of particles, which is determined by the shape and size both. Shape plus size both. So this is not only the size, it is both shape and size, and it is behavior of particles. It is not a molecular weight. Therefore, there is no addition of these words. There is no addition of 60 plus 40. There is no addition of 50 plus 30. So if there is addition, then 50 plus 30 will become 80. But here is no addition, because this is not a molecular weight. This is not only size. It is a behavior of particles in centrifuge machine that is determined by two things, shape and size. Okay, now I think this is understandable for you. It was a big confusion for you, I can understand. Next one is how antibodies stop protein synthesis in prokaryotes. Something like bacteria. When bacteria have attacked in our body, then we are going to destroy that bacteria. Then to destroy that bacteria, we have to stop the protein synthesis of bacteria. When we can stop the protein synthesis of bacteria, then bacteria will die. But how can we stop the protein synthesis in prokaryotes? In bacteria, for example, in bacteria. So for this purpose, we have to stop the production of some these ribosomes. We have to stop the production of some these ribosomes, either it is sorry it is about eukaryotic ribosomes. We have to stop the production of these ribosomes, either it is a say 50s subunit or 30s subunits. There is a certain medicine for different subunit. So let's suppose that we are going to inhibit the synthesis of 50s. So we are going to 50s inhibition. So if we are going to inhibit the synthesis of 50s subunit, then we will use the medicines that is of erythromycin. You can use erythromycin, and you can see chloramphenicol. These two medicines you can use. When we will give these two medicines to the patient, then there will be the no production of 5s subunit 5s ribosomal RNA. When there is a no production of 5s ribosomes unit, then there will be the no production of ribosomes. And you know that what is the function of ribosomes is the production of proteins. When there is a no ribosome, then there is a no protein production. So in this way we can stop the protein synthesis in prokaryotes, and this way we can stop the 30s as well, 30s inhibition. So for 30s inhibition we use two kinds of drugs, that is tetracycline and plus streptomycin as well. We can use streptomycin. So for the inhibition of 30s subunit, we use the tetracycline and streptomycin. So you can see that these are certain and specific for different subunit. For 50s there is erythromycin and chloramphenicol, and for 30s there is a tetracycline and streptomycin. When there is a no formation of these subunits, then there will be the no complete formation of the ribosomes. When there is a no formation of ribosomes, then there is a no production of proteins. When there is a no production of proteins in bacteria and prokaryotes, then prokaryotes will die, and in this way we can cure any disease in our patient. So it was about the ribosomal RNA, that how ribosomal RNA used to construct ribosomes. So in this way they are used to construct the ribosomes, and it was a little bit concept about that S that I was using S once again, that what is about that. And next a little bit break to pharmacology, that how antibodies stop protein synthesis in prokaryotes. So something like that, we can use these antibodies in the form of proteins, that is erythromycin, chloramphenicol, tetracycline and streptomycin. These are the drugs we can use to control the production of proteins in prokaryotes or in bacteria as well. So it was about ribosomal RNA used to construct ribosomes. In next lecture we will discuss about the transfer RNA roles in the translation. So thank you so much guys, see you in the next lecture. If you have any question, then you can ask me, you can comment me, then I will answer you inshallah.